Hello, everyone. Thanks for joining. This is our third of five webinars, part of our annual winter webinar series that takes place around this time every year. Our webinar series is a series of informative webinars we put on starting around Invasive Species Awareness Week. And the goal of this webinar series is to raise awareness around the latest invasive species topics and promote action to prevent their spread. Okay, now just on to our main event. I'm just going to introduce Sarah Kingsbury, our speaker, and then pass it off to her. So Sarah is the Senior Aquatic Invasive Species Biologist for Fisheries and Oceans Canada, working out of Nova Scotia. She specializes in freshwater non-indigenous species, but also has many projects in marine ecosystems. She likes working with data and is familiar with ecosystem-based and species habitat sustainability modeling. Sarah also works with eDNA and genomics, screening level risk assessments, climate matching, and framework development. Mainly, she likes to work on projects that contribute to aquatic invasive species management. And previously, Sarah completed her master's thesis at St. Mary's University in Halifax, studying the Chinese mystery snail. Her Bachelor of Science in Chemistry was completed at the Royal Military College in Kingston, Ontario. So welcome, Sarah. I'm going to end here and stop sharing and pass things off to you.
Hi, thanks so much. Just share my screen and let me know when you can see that.
That looks good.
Okay, great. Thanks so much, everyone, for having me. The title of my talk today is Maritime's Region Watch List, but for those of you who don't live in the DFO or Fisheries and Oceans world, Maritime's region for my program covers um, Nova Scotia. So I am fortunate enough to live and work in Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq people. And some of this watch list work today is also going to cover the provinces of New Brunswick and Prince Edward Island. But primarily my work is focused on Maritimes region, which for me is Nova Scotia. And I'm talking about our two watch list processes. Like Chris said, I have one watch list for marine species and one for freshwater species. And these two projects were done in parallel with each other because they have slightly different target areas or areas of interest for the study. And we wanted to try some different methodologies to do the risk assessment portion of the watch list. And we also have different experts working on these different watch list categories. So that's why there's two separate papers or watch lists that we have, but really it's feeding into the same DFO management team. Both watch lists follow the same objectives and background, which is that we wanted to look at all the many thousands of species that could potentially be introduced to Nova Scotia, and try and filter it down to which species are most problematic for us and try and rank those species based off of how likely we think they are to have some kind of negative ecosystem or socioeconomic impact to our area. My program operates under something called the Aquatic Invasive Species Regulation. So it's a federal regulation that applies across Canada. And it has two sections, parts two and three, where species are specifically listed. And it's difficult to get species listed without first doing this kind of background work to figure out which species should we be trying to list and why, to have a rationale. There's so many thousands and thousands of species and so much biodiversity on planet Earth that it's almost impossible to try and figure out all the species to list. So that's why we needed to do this watch list process. We also, at the same time, had some colleagues in DFO science. So I sit on the management side, and there's a science side in our sister branch. They had developed this framework to create watch lists and try and prioritize species as best we can. But our team is really small. Sometimes, some years we're a team of one person, some years we're a team of five. So we really wanted to see what happens when we try and do this watch list work with our small team, especially in light of the huge mandate that DFO has. We have so many taxa, so different types of fish and different types of snails and mussels and plants that all fall under that sphere of DFO. And we also have a really broad geographic context that we have to plan for. So different types of ecosystems, whether it's marine or freshwater estuarine. So that's a big ask and a big project. It can be a little overwhelming, but we are hoping that this framework would help guide us through that process. And generally, these are the species that we're looking for. So the infographic on the side here really explains well how it all works together. You have a big pool of species globally, but what we're looking for, were species that we thought could live, establish, reproduce in our target area or in our study area that we know have some type of invasion history elsewhere. So we know that they've been problematic to somebody else in some context, um, and that also have a tropical pressure. So that means a way in which humans are bringing the species from where they currently are, or are maybe are native to, and bringing them into our study area or into our area of interest, um, and are actively doing that. So through this work, we were able to kind of filter out that giant list of initial species into a smaller list of species that then we can do a risk assessment process on to rank those species based off of how likely they are to establish, the extent we expect them to be able to establish, and then the extent of those impacts, those negative impacts that I referred to. So I'm going to start with the marine watch list, and I do have slides on the freshwater one afterwards. So I'm going to present them both separately, just because there are some different contexts, especially in the geography of our area of interest. So for the marine side, we were looking at Scotian's Shelf and Bay of Fundy. And you can see highlighted here with that dark blue polygon, that's our area of interest. It's the Maritimes region on our science counterparts boundary, so covers most of Nova Scotia and that southwest portion of New Brunswick. And the reason why we chose it is because it's a really interesting place in terms of biogeography. So there's different types of ecosystem niches there. And there's also a different assemblage of organisms that naturally occur. So it covers three freshwater ecoregions. And if you're new to freshwater ecoregions, they're also referred to as meows, which is just the short form for freshwater ecoregions of the world. And our study area covers three of them, so 40, 39, and 37. And the way that we looked for source areas or areas that might be a source of new species to be introduced to Nova Scotia is we wanted to see which meows had the same or similar salinity and temperature regimes to our study area, and which ones have shipping volume or frequency into our study area, because that would give us both that environmental similarities as like a broad uh, first way of filtering out those lists of the long list of species. And then it also speaks to that tropical pressure. So the human mediated pathways in which species are brought from these other areas into Atlantic Canada. And this is what we're left with when we filter out all of those other marine ecoregions. These are the few ecoregions that are environmentally similar to Nova Scotia and also have that tropical pressure that could introduce new species. The color of the polygon has to do with the number of invasive species lists that are available in each of those areas. So the darker the polygon, the less lists that are available. Some areas that are, bright yellow have quite a few lists available. So overall, we looked at 36 different lists and 2,543 unique species were found across all of these various lists. And when we filtered out the species that maybe don't fall into our DFO mandate, so, pathogens and parasites, our team doesn't cover. We're also more focused on macro aquatic species, so species that you can see with your eyes. Um, and we're also not really interested in birds. That would be a different federal group, so not us. So that left us with 428 species to consider. And when we further assess that list of species, you could see this bar chart kind of looks at that entire, over 2,000 species list. A lot of them, like I said, were kind of excluded because of mandate or environmental mismatch. When we look closer at that species, maybe it was in one of those meow regions, source meow regions, but when we look closer at it, it's really a species that's not going to take in Nova Scotia. Maybe it's like a tropical species that's at the southern range of one of those other polygons. So not really applicable to us in our current environmental context. Some of those species are listed as invasive to other places in the world, but are actually native to Nova Scotia. So those species would have been filtered out. And unfortunately, some species we just don't know enough about them. We're missing information. So those ones we put on a list called data deficient, which means we're hopefully going to be able to reassess them one day when more information becomes available. So in total, we had 192 species that went to the next step after we did all of this kind of big list filtration into a smaller list of species. We had 192 that actually went through to a risk assessment level. And we use the Canadian Marine Invasive Species Screening Tool. There is typically 17 questions in this risk assessment process. Half are dealing with the likelihood of that species to establish, to be able to reproduce, and to be able to disperse from that initial point of introduction. And then the other half is typically dealing with the impact of that species, like in the context of nature and environment impacts, and then also impacts to social structures or economics. We were able to pare down that initial screening level tool to what we called an optimized CMIS. So it only has 7 questions. There was a paper done by Drew et al. In 2017 that had compared the overall like full CMIS assessments to this optimized set of 7, and they were able to determine that these were the 7 questions that really impacted the result of that risk assessment the most, and therefore the ones that you really have to hold on to and answer. And that's what we did here. Each of these questions are scored on a scale of 1 to 3, one being really low likelihood of that ever happening, and then 3 being a high likelihood. And the thing that's really cool about CMIS is that it also takes into account assessor uncertainty and data uncertainty. So if we don't know, oh sorry, this went forward on its own. If we don't know for sure about an answer, we can say that we have a certainty of 1. So low certainty, if we really think we know the answer, we can put a 3, which means we have high certainty. And all of these scores are combined together to give us a point on a plot, but also with bars extending behind it. So here's an example. This species in the center, that is the adjusted CMA score. That's the point. So we're really sure in this context that that species is going to have a high impact, negative impact on recipient communities, and also is very likely to establish if introduced. These bars that extend behind it are the range that that score really exists in, because risk is not really a point on a map. It's more of a spectrum, and it depends on the information available to us. But with these scores, we're able to categorize species based off of where they fall on this scatter plot when we break it into the 4 quadrants. So in this lower left-hand corner, we have the species that we don't think would have large or far-reaching detrimental impacts on recipient communities. And we also don't think they're very likely to establish if introduced. So they're on that low risk category. And then we have the high risk category that I already talked about, that we're sure, they're the species is very likely to have detrimental impacts and likely to establish if introduced. And then we have these two moderate risk categories, which are species that we recommend get reassessed over time, especially as ecosystems change in the context of climate change or in the context of human pressures and development. Whether or not a species falls into or moves from a moderate risk into a high risk is something that could happen with time. But those species are kind of our future planning species that we want to think about again or reassess again in the future. And the species that fall in that high risk category are the ones that we want to plan for now. I didn't mention this when I was explaining the methods, but as kind of a byproduct of this watch list, we were reading a bunch of information and literature about each of the species, and sometimes this information or literature would note what pathways or vectors were associated with that species introduction. So we started writing it down. And every time we would write it down for each of the species, every time it was mentioned. And then when we got to the end of the assessment, we were able to look back and try and summarize at a high scale which vectors are most associated with all of these species. And that can help us direct some of our efforts. The lead author on this project actually went back in and even went so far as to note when a species was, or when a pathway was noted with a species primary introduction, so being brought from one location to a completely new location, or a secondary um, pathway, which means that it would aid in the dissemination of that species after that initial introduction. And then they also noted main. So that just means that that was the main vector that most of the information access agreed was associated with these species. So you can see things like ballast water and hull fouling, contamination, ship transport, all of those types of human-mediated vectors and pathways are strongly associated with these watch list species. And if we wanted to look at some of the highest risk species that were noted through that process, I just threw a couple on the slides. It's nice to have like a visual representation of this. These are just a few of the overall species watch list. Two of these species, rib bryozoa and Japanese wireweed, have since been found in our study area. So um, rib bryozoa, we put out a paper last year, I think, that shows that that species is pretty ubiquitous now across much of Atlantic Canada. And Japanese wireweed was found in Halifax Harbor. So unfortunately, we confirmed that our method worked. We didn't know that these species were going to ever be introduced, of course, when we started our watch list work, but since starting the work and finishing it, we have found two of these species. Okay, so now let's switch gears to the freshwater work. So similar to the marine work, we're looking at Atlantic Canada. So in this case, we were looking at Nova Scotia, Prince Edward Island, excuse me, and New Brunswick. And we wanted to keep this assessment very broad. So we just said any freshwater habitat. We also included inland estuaries because we wanted to include anadromous species since these projects were, happening in parallel. One was marine focused, one was freshwater focused. Some of those species that kind of occur in both might fall through the cracks. So those species ended up being lumped in with this freshwater, with the freshwater work as well. And for global sources, we were looking anywhere that had similar climate to Atlantic Canada. That was really what we wanted. And so we were using a combination of the CABI horizon scanning tool to pull a big list of initial species. And then we also looked at some watch lists from other Canadian provinces, neighboring states, the US GS list, and the US Fish and Wildlife Services list. And we also looked at our own aquatic invasive species regulation because some of those species are listed for certain areas of Canada, but not listed for Atlantic Canada. So we pulled all of that together and had this initial list of over 4,000 species. Um, and we very quickly realized that some of these species were technically noted as included in freshwater habitats or occurred in freshwater habitats, but it could be, a bird that was there occasionally to fish on the lake, but not necessarily something that falls within the mandate of Fisheries and Oceans. So those species got filtered out. We also filtered out any species that wasn't known to be invasive elsewhere. We wanted to really look at species that at least had some known impacts. So we filtered out those species, and then we filtered out species that maybe they had a scientific name change, so they were a synonym with another species in an older list. So those ones were filtered out. And then if there was no way to actually identify that species through current naming conventions of ITIS, which is a naming authority that we use to compare names with. So if we couldn't identify species or their synonyms, those were taken out. So we ended up having a subset of species that we try to do climate matching on. For climate matching, you're looking at gathering all of the global distribution data for that species. Some species on our list didn't have global distribution data, so we're using GBIF to access this geospatial data. Some species didn't have any data available on GBIF, so those ones ended up getting filtered out. Um, and then we did the assessment on comparing where the species currently are found in the world to the climate that's in Nova Scotia and trying to, sorry, in Atlantic Canada, and trying to see how those climates compare. So that led us to a smaller set of species, which is 221 species that ended up going through the risk assessment process. These are species that we know are not indigenous to Nova Scotia, have a likelihood to establish here, are probably going to be introduced through some type of vector or would have the opportunity to be introduced through some type of vector. And so the next step was trying to rank those species based off of a rapid screen level risk assessment protocol called ISAIA. ISAIA is a recognized protocol that's already been used elsewhere. An example would be Belgium. They have used ISAIA to help rank species. It's really quick, 4 questions, very similar to that CMIS protocol I talked about before. There's questions on how likely a species is to establish, reproduce, and disperse, and how and what that dispersal would look like. And then also questions on negative impacts to native species and ecosystem functioning. So those questions are scored between 1 and 3. Same rules as CMIS, 1 being unlikely to happen, 3 being very likely to happen. Unfortunately, ISAIA doesn't have that nice capture of uncertainty as well, but we were able to modify the ISAIA protocol to add in what we call the zero score. And we had zero scores added in whenever we were uncertain about a species, when we didn't have enough information or data to really feel like we could answer that question, we would add a zero and note that as a species that we needed to go back and assess one day in the future when there's more information. So all of these scores get added together and binned into high, moderate, and low risk. So high risk is any species that is scored above a 6. So between a 7 and a 12 would be our high risk category. Anything between a 3 and a 6 then would be moderate, and anything below a 3 would be low risk. And you can see there's quite a few high risk species. We tried to chunk it together by taxa just to provide an overview of all the species that were assessed. Similar to the marine project, we also noted what factors were commonly being referred to or associated with these species. The plot on the side here, I pulled it for the high risk species because I thought that'd be the most interesting for this group to see. So those high risk invaders, again, between 7 and 12 was the risk score. And we can see a lot of those species associated with water gardens um, and recreational fishing, some associated with bilge water and aquarium, and then less with aquaculture and biofouling. And in terms of what species are the highest high risk species, so the species that scored 12 out of a potential 12, um, these are the plants and mollusk and decapod species, and unfortunately, three of them have now been found in our study area. So zebra mussel and Eurasian water milfoil have since been found in New Brunswick, and red swamp crayfish has been found in Nova Scotia. And on the fish side, walleye are have been found in New Brunswick. So again, a unfortunate validation of our protocol. I did want to include quickly just a small section on implementation because as a manager, the taking of science and projects and putting it into on the ground action is the most exciting part of our work. So why should you maybe consider doing your own watch list process? Well, for us, it's been extremely beneficial. We've been partnering closely with our local chapter of Invasive Canada, which is called the Nova Scotia Invasive Species Council, to get more community groups aware of the species that are currently here, and then also those watch list species. And we actually have a project with our local chapter, our council, to allow community groups access to freshwater habitat monitoring. So some of this is through collection of environmental DNA. We provide the training, the resources and the tools, and then also coordinate all of the lab analyses. And these community groups, which are from across Nova Scotia, have been able to go out and survey and monitor sites that are important to them for their own reasons, and they don't have to tell us why they're important, although I love to learn about it. So it's a really collaborative and autonomous process for those community groups and a great partnership for DFO to expand our sphere of influence and get information out there about these species. We've also been able to create new reference materials for identification guides, or I put in here International Barcode of Life. If people work in genomics at all, you probably know that BOLD, that's the short form for that repository, is just a big repository of genetic sequences for each of these species that are publicly available or mostly publicly available. And so we have been able to start working towards sequencing the species that are on our watch list and looking into new ways to improve the genetic identification portion of these species. On a broader scale, we've been able to really focus our efforts. Like I said, we're a super small team. So we've been able to focus our efforts on pathways instead of dealing with species individually. And I mean, often after things become introduced as well as when we see action taken. But because we now know that there's these pathways associated with each of these species, and we can kind of look at this from a zoomed out lens and explain to people and really have this great background of information to show with them and share with them and say, we're looking into this pathway or vector that affects you, but here's why, and here's all the species associated with it. And that's why we're really concerned about it. And so it's a very transparent and collaborative process with the industries that are impacted by our work as well. And I think this might be my last slide, but I just wanted to say that our goal in management is always to work towards this integrated management approach. So we want to identify species that are currently here or are on our watch list and look at where we think they're going to be introduced and how likely that habitat is to be suitable for that species to establish. And then look at what pathways are bringing those species into those sites. And so we can really get away from controlling non-indigenous species that are well established throughout our range and working more towards that pathway. So less of this species focus, or I guess dealing with things after they've arrived and more of that prevention work and dealing with the humans and the human populations that are moving species around and introducing them. We also wanted to get towards or away from this term of priority areas. That's very popular in management, and we certainly use it too. So we're not immune to this term of priority areas. But when you say priority areas, we often hear from people, priority for who and for what. And so through some of this climate matching work and the meow assessments that for the marine work, we were able to start looking at areas that we need to be surveying more closely or surveilling more closely, and that could be because of the vectors that are active in that area, or because there are areas that we think would have more climate matching, more species that would find suitable climate there, and then also areas that are identified for conservation focus. So where we don't yet have invasions and we want to protect them from invasions, looking at those areas and being able to tell them these are the pathways that we're most worried about. Are these in your area? If so, what can we do to try and work together to mitigate that problem before or mitigate that pathway before it becomes an issue? And then the last thing, we had originally been making response plans. I think some regions call them early detection and rapid response or EDRR plans. In our region, we just call them response plans. But we were making those based off of species. And then we started noticing between species of similar taxa, the plans didn't change that much. Our actions when we're responding to a new detection of a species were broadly the same. So we started moving towards this taxa-based response. And because we have the watch list, now we can be proactive in including those watch list species in these taxa-based response plans. So instead of having a response plan for smallmouth bass, and then another one for chain pickerel, and then another one for, the next species maybe on our watch list, we could just include all of those in one plan and have them all listed there. If these species show up, here's our concern, here's the risk assessments already done, and here's the actions we expect to do. So that's all I really have for you. I did include the two paper titles and author list here. They're both publicly accessible. They're published in open access data, so anyone should be able to access them at any time. And that's it. Thank you so much.